Hello, thank you for coming to the internal cycle of seminars today. As most of you know, today we have two important events. One is the seminar of Samuel, and then we have a celebration for the promotion of Nuria Marva and Ramon Chapeiro, two professors of investigation. So now, first, Samuel is going to present a little part of his work that he's doing here. He's in the Global Change group, working with some CO2 things. Exactly. It's serious meadows, with her supervisor, that is Hendrix. And thank you so much for collaborating in the seminars, and go ahead with the talk. Thank you.
Hi everyone, thank you very much for coming to this seminar. And yes, the opposite. No, it's not working. Because you have to click.
I know.
It's working. Okay. Hi again. Thank you for coming. So today we'll talk about the current and potential future CO2 dynamics in seagrass meadows. I'm a student from the European Institute of Marine Science from Brest in marine biology. But here I am doing biogeochemistry, which is the science of we are looking at the processes that governs the composition of our environment. So it means we have to look at all the spheres, like the biospheres, the hydrospheres, the geospheres. It's a very disciplinary science. So sometimes I don't know if I'm a biologist, if I'm a chemist or a physicist, but I'm having fun. So that's the most important part. And I am looking at CO2, carbon dioxide, which is like the building block of all organisms, and CO2 is transformed with photosynthesis into organic matter and O2. But in recent history, there is an explosion of the CO2 emissions due to human activity. And we went from 5 billion tons in the 50s from 35 billion, which is a lot. And the thing is that CO2 is also a greenhouse gas. So the temperature is rising as much as CO2 is rising. And we have different scenarios depending on the climate policies we are doing. And the current policies, if we respect the policies, temperature will only rise at like 3 degrees, but if we don't do anything, it will be this. But this week in France, they are saying that they are already expecting temperature to rise by 4 degrees and they are now agreeing on this. So that's a nice time to be alive. But where does it come from and what can we do? Carbon dioxide mostly comes from the emissions from the burning of fossil fuels, and carbon dioxide, half of the carbon dioxide stays in the atmosphere, and 58% is either taken by the plants or dissolved in the ocean. And you can see the consequence of this. If you look at the CO2 time series, it's been this time. Sorry, you can see that almost ppm. This is the CO2 monitor. Like this is the part per million. There is 100 more now than in 60 years inside the atmosphere, and the pH is also rising, and is also sorry, it's the opposite. pH is going down. Sorry, I'm very stressed. I'm not used to talk to as many people. Anyway, the partial pressure of CO2, that's the CO2 that is dissolved inside the ocean, is rising a lot. And yes, blue carbon is the carbon that is sequestrated by the seagrass, the salt marsh, and the mangrove, all the aquatic ecosystem. And this is long-term storage, which means that short-term storage is made by the phytoplankton, but it is not trapped. But Posidonia is trapping the CO2. And since coastal meadows are long-term carbon sinks, this is very interesting for the policymakers that are interested to know how much coastal meadows can mitigate climate change by removing CO2. But it is our duty as scientists to translate our knowledge into something that policymakers can understand, and they understand money. So we must assess the natural capital value of seagrass meadows, ecosystemic services. But for that, we need to know how much they can uptake, how much they can remove, or also how much they produce. So my goal in this internship was to investigate the dynamics of CO2 and associated variables in the coastal areas of Majorca, and also the role of seagrass in the CO2 uptakes. And for this, I did some time series analysis, and I made a surface chamber to measure the air-sea surface transfer. And also I want to quantify the impact of global changes and thermal stress on seagrass meadows mitigation potential. So here in Medea, we are constructing biogeochemical time series datasets for a long time now. And I am using data from 3 different sites. One is the Bay of Santa Maria in Cabrera, which is a very pristine site. So there is no it's inaccessible for the human activities. So it's very non-polluted. And we have the oceanographic bureau in the Palmas Bay. And one that was very interesting for me was the Salines Lighter Coastal Research Station in Salines, where we have a bimensual sampling since 2008 of physical parameters, chemical parameters, and biological parameters. But this is a lot of parameters. So how can we analyze a large number of variables, and how can we look at the correlation? And one tool to do this, because it's a lot of dimensions, it's like what we can do is to try to reduce the number of dimensions, try to squeeze them together. And you can use principal component analysis, which is like if you take a 3D object and you look at the shadow, this is like a projection, and you can look at different ways to turn the object to find the best projection with the most variation. And that's what I did with all my variables. So one point is like one sampling day, and it's projected through 2 axes with the most variance explained. And here, with my analysis, we can see that more than 50% of the variation is explained by those 2 axes, which is very good. But this doesn't make much sense like this. So I have to, because this is very abstract, like this line or this line, and we have to identify what it would mean. And what I did is I looked at the season where the point was taken, and you can see that it's more winter here. This is summer this way, and autumn and spring is in the middle. So you can see seasonal pattern like this. And also, if you do the same with years, you can see that points are more up as long as time goes. And so my idea was to put names on the, this is more past and this is more recent. And here is summer and winter. And you can, what you can say is that the partial pressure of water and the temperature is higher in summer. This is more correlated. You can make some correlation. This is very interesting because you can do this with much higher database. And okay, we did another analysis with a much longer time scale on the CO2 inside water, and you can see a seasonal trend. And what we wanted to do is to estimate the trend of how much it's rising every year. So we did a sinusoidal regression, and we found that the mean CO2 raise is the 4.46 ppm per year, which is twice as more as in Lampedusa. So it means that in Majorca, the rise of CO2 is higher than in other places. And also, the optimal period is supposed to be 1 year because the season is like this, but it's shortening. This is also interesting. It means that there is a change. And also, we can use physical laws to try to estimate what parameters will influence the CO2 inside the water. And Henry laws means that you can use Henry laws, which says that the CO2 inside the water is a function of the CO2 of the air and the temperature. And if you reconstruct the data set like this, you can see it's going pretty well together. So we can say that those 2 parameters seems to be the main drivers of CO2 inside the water. And for the CO2 to go from the air to the water, it needs to be transferred. And it's very interesting to look at transfer rate. And for this, you can take a, okay, sorry. So the flux, this is what we want to measure, is the quantity of CO2 passing through a surface unit by time. And with this, we can measure the speed at which CO2 is going inside the sea. And the problem with this is that it's not easy to measure. And we can make in situ measurements, or we can use empirical model. This one uses wind speed. And it's interesting to look at the different results between the two methods. So this is why I built a surface chamber that will, so I can measure the difference and try to compare the data. So like we tested them, we tested the machine, that's me. And the thing is that the construction phase was okay, the testing phase also, but then the gas analyzer broke, so we couldn't do much field measurements. So I was left with only some test measurements, and still I have some. I was able to measure some transfer coefficient, velocity, but you can see that they are very different. So we have a significant difference between the estimation of K. There is not one that is right or wrong, it's just different. But this is promising, and it just needs to be redone when we will have the material again. And so this is my last part, it's more biology. So Posidonia seagrass, they mitigate carbon, they are uptaking CO2 from the atmosphere by photosynthesis, but they also release it with respiration. And when they undergo some pressure, some damage, what happens is that they release more CO2 than they can uptake. But what happens when they can suffer a lot of heat waves are more and more frequent in the ocean. So this is we need to know how they will react to this kind of stress. And my goal was to quantify the impact of heat waves on Posidonia oceanica and how it will impact the carbon sequestration potential. And for this, I made some climate chamber experiments. So we incubated some seagrass in a climate chamber where you can control all the parameters. We applied a thermal stress for a month, and then we measured the sampling of, we measure and sample some key metabolic parameters. So this is I had like each pore has one Posidonia, some shoots of Posidonia, and some of them only are Majorca, which is only the shoots. No, there is no leaves anymore. And what I did is I had two tanks. One was always after 15 degrees every day, and we incubated them. And another one I was gradually raising the temperature. And then for each incubation, I did some incubation for 24 hours. Where before and after incubation, I was measuring the salinity, the greenhouse gas inside the water, and the total alkalinity. And during the incubation, we have some sensor that we put that every 15 minutes measures the pH, the oxygen, and with this we can calculate the CO2 inside. And by the end of the experiment, we also looked at the composition of the plant, also the leaf necrosis. That gives us a good estimate of how stressed the plant was. So we took the cores in the Cala of Santa Maria in Cabrera in March. And then this is what my experiment looked like. So the sensor, for 24 hours, they were measuring the O2, and you have so they are producing oxygen in the plants, and that's what you can see. It goes up, and during the night there is no photosynthesis, so it goes down. And you can measure the net production of O2 like this. You can look at the end and we start. Then the respiration, is like the opposite for when it's night, and you can evaluate the gross production like this. And for CO2, it's the same. You can evaluate how much CO2 is sequestrated by looking at how much it was CO2 there was at the beginning and how much there was at the end. And of course, during the night, they are emitting more CO2 because they are not taking it anymore. And those are my results. So we can see that this is the control. So it was not heated. And you can see that there is more starch inside the control course, the control Posidonia than the heated one. And this is the opposite for saccharose. And starch is like a sugar that is decomposed when the plant is stressed to produce saccharose and fast sugar. So it would mean that the plants were very stressed when it was heated, and also necrosis rate was much higher during the heating. So these are the oxygen profiles from my course. So this is different days, and we did 6 incubation, and so you can see the up down, and you can see that the profiles are significantly constant for the control course. So it means that there is no effect of the manipulation on the results. And this is not the case for the heated course. You can see it's going downwards. And at the end, one of them died. So that's why there is almost no oxygen production. But you can see decomposition here. Anyway, for dead matter, it's the same behavior, only it's a little bit less. And the productivity of our respiration ratio is very interesting for us to know how much they produce O2 in comparison to the respiration. And you can see that between when it's heated, the Posidonia, the productivity of Posidonia is going way down after heating. But dead matter is not affected by the thermostat, seems so. And also you can see that this is different temperature. This is the highest temperature, and the difference of CO2. This is like the uptake of CO2 during the day. So you can see they are taking less CO2 when it's very hot, and also the release of carbon during the night. This is negative, but it's more. So they are emitting way more CO2 when they are heated. But also in my experiment, I have some problems with the sensor and the salinity. So maybe there's an impact of salinity in my results. But what we saw in my analysis is that Majorca, the dissolved CO2 is going up at higher rates than other places in the Mediterranean Sea. So now it's up to us to know why. And also there is a significant loss of carbon uptake with thermal stress, and which implies that the capital value of seagrass meadows will be affected by climate change. And I tried to put a value, an economic value on this. This is a very recent article that is trying to put money value, capital value on Posidonia, and it says that in the Western Mediterranean Sea, it's 2.28 euros per square meter per year, and so it's more than 6 billion euros for every Posidonia. And what I did is I estimated that it was 80%, 20% less efficient when there was a heat wave, and the impact would be that the daily capital loss of one heat wave per day, it would be 600,000 euros per day of heat wave, which is a lot, but I don't know. It's just like it's a big model, so I don't know. It's the difference between reality and model, maybe a lot, but it's still a good estimation. I don't know. To me, it's what we have to do, you know. So okay, that was difficult for me, and maybe for you. I'm sorry, but thank you for your attention. And I always ask you questions, like stupid questions. So now it's your turn. Yes.
I have a couple of questions. One is in the model where you assess the transfer of CO2 across the season phase. You saw an image where the model was predicting.
Yes, based on wind speed.
Yeah, yes, and temperature, I think it was, no, it was
Yes.
CO2 concentration and temperature.
Because the sheet number, I think it depends on the temperature.
And it surprised me that the model was the minimum and the maximum were lower than the two values.
Yes, I think
Why do you think it's occurring there?
I can't really say. To me, I don't trust this value too much. I will not make too many conclusions. But to measure the wind speed is quite difficult when you are on the boat because you can just go like this, and you're supposed to go at 10 meters, so you have to make estimations after. So the wind speed is not very precise, and which has a huge impact because it's squared. So when it's below 1, changes a lot. Also, I had to estimate further. So, yes.
Well, still, I understand your. But anyway, I have another question about the experiment.
Yes.
Which I didn't understand very well because, well, one thing is that you have done the experiment now, I suppose.
Yes.
And you have done a temperature treatment with some of the plants during 1 month, or yeah, something like that. So now you have used in the experiment Posidonia that this was on winter time, and you have created a heat wave, no?
Yes.
That's
In
really
April.
Do you think that if you would have done this in summer, you would have a different response?
Yes. Totally.
Considering the seasonal adaptation that
Yes.
plants in nature face, because this would be a really very big heat wave for a winter plant to suffer and increase to 30 degrees. No?
Yes, but also we are expecting the heat wave to be more violent even in summer. So, but it's true that in April the leaves are also shorter, and since they are not used to that kind of climate in April, it's true that maybe there is some derivation from what could happen during actual heat waves. But yes, I think a second experiment would be to do the same, but with shoots taken in summer. This is true.
Thank
you.
So the changes in CO2
Yes.
uptake, is that a relative CO2 uptake, meaning,
Yes.
or is it absolute CO2 uptake? So it takes into account the fact that different temperatures have different solubility of the
Yes.
of CO2.
We have different solubilities, but I assumed I took the solubility at 20 degrees. I didn't take a different solubility each time. I know I should, but
How much does do you know how much the solubility changes between, let's say, you try 15 and 30 degrees? I mean, I don't know, 1%, 5%,
yes,
10%.
it was not. I checked, and to me it was not that much. But yes, that's what I neglected.
Because at high temperature, you will, I guess, have more CO2 dissolved in the water.
Yes, it's like
so therefore the absorption is even smaller than the one that you estimate using the solubility at 20 degrees.
Yes. So you think I should take it into account?
Yeah, I think it's, yeah. I think if you know what the solubility is, which I bet you do, then
okay. Yes. I'll do that.
I'm not an expert in seagrasses, but as far as I understand, is you try to relate these with climate change, and but most of what you're measuring, I didn't understand wrong, is it's actually carbon fixation, is primary production in some sense, but not carbon sequestration. Actually, carbon sequestration is how much of all that carbon fixation is long term, is good for long term.
Yes.
Because otherwise we have not any influence on climate budgets. So how much does Posidonia actually bury of that carbon that is fixing?
Well,
Do you have an idea of that?
it takes more time, of course, to get sequestrated. And yes, I should have stressed that in the end, my calculation, for example, I put some values that are in tons of carbon removal, but this is just taken from the hydrosphere or atmosphere, but it's not going inside the sediments. That is true. But still, it's in the process, and it's much more sequestrated than, for example, phytoplankton. And to give you a percentage of how much goes, it depends a lot on the physical parameters, spatiotemporal parameters. So, yes.
Yeah, of course, it sequestrates more than phytoplankton. And the thing is that phytoplankton is over 70% of the globe,
Yes.
whereas this is a very thin stripe on the coast, and that's one thing. And it's even more subject to coastal effects. I don't have that number, but how much actually gets buried for a long time, which is, I think, is the relevant part of for CO2 budgets, general budgets.
I was curious, I just remember there's another thing that I wanted to ask you about the experiments, which is the amount of CO2 in the water, I guess, because you're using a small enclosure, right? Will this depend also on the microbial activity inside?
Yes.
So are you taking into account what happens there? Because, for example, if you increase the temperature,
Yes.
I guess you're going to have a higher production of CO2 by the microbial communities inside the water.
That's what I.
In particular, if you start to have necrosis, right, solution of the organic matter.
What I see is that this is CO2 produced by dead matter. So it's to me it's like microorganisms because it's shoots of Posidonia but without the leaves. So it was a way to investigate this exactly. And maybe we can subtract and see what's left, and that would be a possibility. But I saw that well, the difference is not. You can see it's pretty stable for this enclosure. So, and here also you can see that the productivity is very similar. But that's what I would say. It doesn't affect, which is surprising because yeah, if you hit the microorganisms, usually they more.
but if you have the leaves there, maybe when you increase the temperature, then the leaves can degrade and then generate nutrients that can provide a substrate for the microorganisms to grow, which you won't have with your dead matter.
Yes. Thank you. Yes.
Another way of showing this graph, because this is all it's positive, no? So
Yes.
What is which are the primary producers in the dead matter? Because you have no
Yes.
it's like you have production,
We have-
production.
We do, yes. But the thing is that it's not affected by temperature wise. That's what, because it's the same.
I think it would be more informative if you, instead of just grouping it by control and temperature, to see across your temperature treatments, because we have like many treatments, no?
Yes, but
How this varies across, not just pulling all the temperature increase together. But yeah, actually you have a substantial production too.
yes, it is substantial, and it's actually it's also surprising to me. But what is interesting is like the reduction for Posidonia. It's true that the difference is not as big. I would have expected to be much more different because we have photosynthesis here. Well, much less.
Well, so this is a ratio. So I don't know how the absolute values of gross primary production or respiration.
I didn't put that. Yes.
Probably there are
Not about experiment, but do you know which parameters are considered in the capital loss? Like, just as a curiosity in the study, because you have not only all the things about, I don't know, carbon storage, etc., but also like coastal protection. We say
Yes.
like if this number takes account like
No,
everything.
I did this.
No, but not yours. I mean, the one in the, I mean, the people who study this. Do you know in the paper
Yes, but
which parameters they?
They consider all the ecosystemic services, and CO2 removal is only one ecosystemic services. Posidonias are a nursery for fish, for example, fishes, other species. They are protecting us from the waves, from the tidal, well, from the waves. So they are trying to give a value. It's like if it wasn't here, how much money we should put to have the same effect, but because that's the idea behind the quantifying with money. Yeah.
Okay.
Yes. This is also why it's interesting to quantify exactly what happens if there is a heat wave, how much carbon is not removed by Posidonia. That was the. That's it.
And so the temperature that you set is that of a heat wave, because I guess that here we have two effects. Now you have that on average the temperature rise.
Yes.
So you can ask yourself, how does the Posidonia react to a constant change of temperature with climate change, or to a pulse, I think so, to a short time, but with a much higher temperature. So the temperature that you set is that of a heat wave, or like?
Yes.
Okay.
It's over 28 degrees. You consider like the metabolic activity of Posidonia is seriously damaged.
Okay, so last year we've been on a constant heat wave, essentially, right? On the
You can measure the number of days of heat waves, and
It was like a 3 week.
so imagine like €500,000 every day. That's in the calculation. So this is substantial.
No more questions?
Yeah.
Okay.
Yes. Sorry again, I was super stressed. I didn't say what I wanted.